Hello there. Today's topic is specialized tissues of the heart and the nerve innervation. Before we go into the topic of discussion, let's refresh our memory regarding automaticity and rhythmicity, which was discussed in the previous video. What does automaticity and rhythmicity mean? It means that even the heart is totally denervated. The heart has the capacity to initiate its own beat, called as automaticity, and beats at very regular intervals, called as rhythmicity. Why the cardiac muscles have these two unique properties? These two properties are caused by specialized pacemaker tissues of the heart, consisting of the sinoatrial, SA node, and atrioventricular, AV node, the bundle of His and Purkinje system. Rhythmicity of the heart is due to automatic discharge of cardiac impulses at regular intervals. Normally, the SA node acts as the pacemaker of the heart, which means its impulse discharge determines the rate at which the heart beats. The cardiac impulses generated in the SA node spread to the atrial muscle, and little later, through the bundle of His and Purkinje tissue, to the ventricles. The pacemaker tissues other than the SA node normally act as conductive tissues only, even though they have the capacity to generate impulses on their own. The reason why normally they do not do so is that the intrinsic rhythmicity of the SA node is far greater than the intrinsic rhythmicity of other pacemaker tissues. Therefore, other pacemaker tissues receive an impulse arising from SA node before they can generate their own impulse. Let us go a bit deeper into the nodes. We will start with the SA node and continue with the AV node. The sinoatrial node is a small strip of specialized cardiac muscle tissue located in the right atrial wall immediately anterior and lateral to the opening of the superior vena cava. Located in the center of SA node are still smaller modified cardiac muscle cells called the stellate cells or P cells. P cells are believed to be the real pacemaker cells. The SA node also contains parasympathetic autonomic ganglia and postganglionic sympathetic and parasympathetic fibers. The pacemaker activity of P cells is modulated by the activity of autonomic nerve fibers. The AV node is located in the wall of right atrium at the right posterior portion of the interatrial septum. It consists of specialized muscle fibers resembling those of the SA node, including the P cells. The AV node is connected to the bundle of His through modified cardiac muscle cells called the transitional cells. The bundle of His is the only electrical link between the atrial muscle mass and the ventricular muscle mass. Therefore, the AV node acts as an electrical gate. Cardiac impulse arising from the SA node spreads all over the atrial muscle mass but has to pass through the AV node before transmission to the ventricular muscle mass. At the AV node, there is always a delay of about 200 milliseconds before the impulse is transmitted to the bundle of His. Another important property of AV node is that it is not capable of transmitting impulses at a very rapid rate. If pathologically, the atria start firing impulses at a rate of 200 impulses per second, then the AV node allows only some of these to pass into the ventricles, which means the ventricles are not allowed to beat at very rapid rates. This is physiologically useful since very high rates of ventricular contraction make it ineffective as a pump. Besides these two important nodes of the heart, there are organized system that connects the nodes and the cardiac muscles called as His Purkinje system. The Purkinje system consists of bundle of His and its branches. It contains muscle fibers larger than the typical ventricular muscle fibers. These fibers are striated but do not have distinct cell boundaries. Their conduction velocity is also faster than the typical myocardial fiber. The faster conduction velocity helps to spread the wave of excitation rapidly over the two ventricles, so that the two ventricles contract simultaneously. The bundle of His begins from the AV node, passes through the fibrous AV ring into the top of interventricular septum. Almost immediately it divides into the right and left bundle branches, which supply the right and the left ventricles, respectively. The left bundle branch further divides into anterior and posterior fascicles. The anterior branch is thinner of the two and supplies the anterior wall of the left ventricle. The thicker posterior branch supplies the posterolateral wall of the left ventricle. The right branch and the two fascicles of the left branch run subendocardially on the respective sides of the septum. Now we will discuss about the innervation of the heart. The heart is innervated by both the sympathetic, as well as the parasympathetic nerves. The autonomic nerves regulate the frequency at which the heart beats, and the force of each beat. Changes in the discharge rate of autonomic nerves to the heart help to adjust the cardiac function appropriate to the various stressful states the body is subjected to. The parasympathetic fibers to the SA node are supplied mainly by the right vagus nerve and to the AV node by the left vagus nerve. Stimulation of the vagus produces decreased heart rate and decreased force of atrial contraction. The cardiac output is decreased only moderately because of decrease in the heart rate. The sympathetic fibers innervate all parts of the heart, including the SA node, AV node, his Purkinje tissue, atrial and ventricular musculature. Stimulation of sympathetic fibers to the heart produces increased heart rate called as positive chronotropic effect as well as increased force of contraction of the atria and the ventricles called as positive ionotropic effect. As a result, the output of the heart is markedly increased. These are some questions that can test your remembrance of the topic. Pause this video and try to answer them. That's all guys regarding specialized tissues of the heart and its innervation. If you find value in this video, please like, comment, and subscribe. If you have any biology topics that need discussion, send them via the Google form that I have linked below. Thank you guys and see you in the next video.